talk to you about something called the complement system. Now, all the immunologists that are watching, if there are any, would be like, he's not going to go through the complement system. And no, I'm not, because it's too big, but we're going to do a very, very quick overview. And the complement system is fundamentally a series of proteins that are in your circulation. And they exist in what's called the complement cascade. So a cascade, as I'll show you, is a very complex series of activations where one protein activates another protein that goes on and activates another protein that goes on and activates another protein, and so on and so forth. But the goal of the complement system is to form something called a MAC, which stands for membrane attack complex. And the membrane attack complex, what it does to invading bacteria is it forms a pore. In the membrane of the bacteria. Which you can loosely think of as a hole. So if you've got your bacteria here, your MAC looks like this. So this is your MAC. It's a hole that's been punched in your bacteria. And what, unfortunately for the bacteria point of view, the innards of the bacteria leak out of these holes. And it causes death to the bacteria. Which is good, because you don't want bacteria floating around your circulation. So let's have a look at the complement system from a bit more detail. I'm not going to draw it because you're going to see how complicated it is. So, right. This is what I mean by a cascade. Give me one sec. Get this any bigger? Maybe we can't get this any bigger. But it all starts with certain molecules that bind the surface of bacteria. Okay? And then those molecules that are bound the bacterial surface, what they do is they recruit other molecules that then get activated, and then those recruit other molecules that get activated. And there's an amplification loop in here as well, which means that you only need a small kind of spark to light the touch paper, and then everything kind of cascades on its own. And after a certain amount of time and a certain amount of molecules kind of converting and activating other molecules, you end up with this MAC complex down here. This is the surface of a bacteria, if you can see, that has several MAC complexes punched in it. So this is an electron microscope image, top down, and you can see that there's loads of holes in this surface, and presumably the insides of the bacteria are kind of leaching out of these holes. Very nasty for the bacteria. The triggers, because you don't want this complex, firing off willy-nilly, you don't want it affecting your own cells. So the immune system has evolved a complex series of triggers for the complement system. So if you imagine this is the bacterial cell surface, so the membrane. And this is inside. This is outside. You have two kind of classical triggers, if you like. There is a third trigger, but we won't go into what it is. So you have sugars. They're called sugar residues, and they are part of the bacterial cell wall. So I'm going to draw these in. This is just an innate kind of, bacteria just have these sugars on their cell walls. Human cells do not, though. So this is the good differentiator between why the MAC complex only forms on bacteria and not on humans. You have a sugar called mannose. I think it's double N. And the complement cascade is triggered by a molecule, a protein within the cascade called mannose binding lectin. Lectin is another protein, type of protein. But what this can do is this can bind the mannose sugars on the surface. This is mannose binding lectin. Okay. That's part of the complement cascade. You can also get complement activating via the binding of an antibody. So let's draw an antibody. A bit thicker. Let's draw an antibody. Like so. And both of these structures can trigger this cascade. Then what you'll get is protein after protein after protein activating. And we represent that, say, I'm only going to call this, I'm not going to give the actual numbers, but say that is C, and that is, anything with C on it is just a complement cascade. What you get is that will convert this into two things, which will convert that into another two things, this kind of thing. This is the way it kind of goes. And it goes so on and so forth. This is exactly the same thing. You get conversion of one protein into another one, or splitting in half. And then so on and so forth. Until eventually you get the formation of the MAC. This all kind of results in the MAC formation. Remember, this is all still on the surface of a bacteria. So then let's extend the surface a little bit out a bit. Just like so. And just drawing that, what will eventually lead to formation of the MAC, which we'll draw in gold because it's just like we did before, where you'll get a subunit entering and then another subunit entering, and what happens is that eventually forms a pore that you can look down, and the guts of the bacteria spew out. And that is a prime example of a system within the innate immune system that is indiscriminate about what type of bacteria it will target. It will target anything that has mannose on the surface or has an antibody bound to the surface of it.